Since the beginning of human thought, we have tried to explain our world and all the forms of life which inhabit it. We have created myths and legends to describe our origins and our destiny. We have sought order and meaning through belief in a divine plan for the universe. And we have turned to science to describe the evolution of all life on Earth, to explain a system in which the biologically and mentally adaptable survive the changes wrought by time. To a large extent, our knowledge and understanding have expanded throughout the centuries. But no matter how much we learn, the mystery of life is yet to be fully unraveled, and the final answers are still elusive. Perhaps no greater mystery exists than that of human life. A newborn child, sex, female, weight, 7.5 pounds, height, 20 inches. A footprint and handprint are made to acknowledge her as a unique individual, but family speculations already link her to her biological heritage. Should we name her after her grandmother? Does she have her mother's eyes? Her father's temperament? Is that her uncle's chin? Over a century ago, a shy, quiet Augustinian monk performed experiments which provided a degree of precision to such speculations. Father Gregor Mendel, a priest, teacher of biology, and within the walls of the monastery of Bern, an investigator, chose the common garden pea as a subject for study. Mendel conducted experiments which have revolutionized our understanding of heredity, or how traits are passed down from generation to generation. After 2 years of testing to ensure the purity of his samples, Mendel chose 22 kinds of peas with clearly defined characteristics. These were the basis of his experiments in hybridization, the crossbreeding of plants with different characteristics. In his first round of experiments, Mendel set out to determine the results of mating round with yellow peas and yellow with green. To find answers, Mendel planted the purebred peas in his monastery garden. When the plants blossomed, he hand-pollinated round with angular peas, green with yellow. Mendel was like a detective looking for a clue, taking great time and patience to observe how each single characteristic was carried over into the offspring. From the crossing of purebred round and angular peas, Mendel observed only round offspring. Was the round trait dominant? From the crossing of purebred yellow and green peas, only yellow offspring resulted. Could yellow be the dominant color? In repeated experiments, Mendel saw that almost invariably, purebred parents of different shapes produced only round peas, strengthening his theory of dominant and recessive traits. Yet when Mendel allowed the first generation offspring to self-pollinate, as they would naturally in the field, he found that the dominant traits gave way somewhat, and that the recessive traits made a reappearance in the second generation. For example,
可是在
when Mendel separated peas from his experimental second-generation plants, he found a larger total number of round rather than angular. This was not always the case with individual pods, for some pods would contain all round peas, others all angular. Overall, though, round peas of the second generation outnumbered the angular 3 to 1. Mendel's first contribution was that two purebred parents with different physical characteristics will always produce a first, or F1 generation, displaying only the dominant trait. And his experiments with succeeding generations shows that the recessive traits reappear, but only at the rate of 1 for every 3 instances of the dominant trait. We can see how this works if we follow the genes of the parents. 2 large R's represent the round genes, 2 small r's the angular. When the parents are crossed, the genes separate and rearrange themselves in the F1 offspring. The round shape will prevail in all F1 offspring of purebred parents, even though the angular gene is present. But with the mating of F1 parents, we give the recessive trait a chance to reappear, as the genes separate into 4 possible combinations in the offspring. Three of the offspring in the F2 generation have the dominant gene and are round. The 4th has only the recessive and is angular, bearing out Mendel's observation that dominant and recessive traits are mixed in a 3 to 1 ratio from the first generation on. Mendel further found that by a backward crossbreeding between plants of F1 generation and purebred parent plants showing the recessive trait, offspring were about evenly divided between round and angular. Mendel chose F1 plants with both dominant and recessive genes, and parent plants with only recessive genes. He calculated that the genes would group themselves so that the offspring of this backward cross would show the dominant and recessive equally. His careful breeding and mathematical analysis proved once again that gene distribution and dominance were no accident. But what would happen when combinations of dominant and recessive traits were crossbred? Again, Mendel laboriously hand-pollinated his experimental plants and waited for the peas to mature. For this experiment, he used purebred parents with yellow angular and green round characteristics. The resulting offspring again convinced Mendel of the uniform dominance of yellow and round in the F1 generation. The F2 generation was carefully crossed and raised in Mendel's usual patient way. The F2 generation showed all 4 possible combinations of color and shape, which implied that each trait was passed on independently of the other. And just as he found with peas exhibiting a single characteristic, Mendel observed the same 3 to 1 ratio of dominant traits to recessive. Scientists before had casually noted these combinations, but Mendel was the first to anticipate, observe, and record the almost constant mathematical ratio. Today we know that Mendel's findings in the pea experiments apply to heredity in people. By establishing that parents have a given combination of dominant and recessive traits, we know the approximate odds that each of their children will inherit a particular color of hair, color of eyes, or other physical and mental capabilities. Mendel's work was an extension of the findings of other biologists who used microscopes to discover that all living things were composed of cells, and that the cells themselves had an independent life. The anatomy of the cell itself was revealed. In every cell, the dominant feature was the presence of a nucleus, the reproductive center of the cell. Another startling discovery: reproductive cells divide themselves in two, a process called mitosis. Here's what takes place when a living cell divides. Clusters of tiny sausage-like rods called chromosomes gather near the center of the parent cell. Soon the chromosomes begin to split lengthwise. The split chromosomes then move to opposite sides of the cell. An elongated cell then becomes pinched off at the center and forms two new separate cells. But what of an entirely new life? Here, reproduction is the result not of a single cell splitting into two, but rather of two cells, the sperm and the egg, joining into one. The result, a fertilized egg. Each of the billions upon billions of cells that develop thereafter would contain the identical traits and characteristics that had been passed on from the original sperm and egg. But how does the fertilized egg develop into an organism that bears distinct similarity to its parents? And within a species, what makes some of us tall, some short, some big-boned and others small-boned, some light-skinned and others dark, some blonde, some brunette? What accounts for all of these, and thousands upon thousands of other traits, which make us distinct and distinguishable from one another? Geneticists are discovering some of the answers deep within the chromosomes. Each chromosome carries a special group of complex chemical substances called genes. There may be tens of thousands of genes strung out like tiny beads along a single chromosome. In these genes is heredity information, the genetic code that will determine an organism's size, color, shape, and sex, the thousands of ways its characteristics will have been determined by its parents. The code is carried by a remarkable molecule called DNA, deoxyribonucleic acid, which exists in each gene. Here, in this huge model, is a scientist's version of what a single, long, twisted DNA molecule looks like. These molecules are like computer tapes containing coded messages that will be transmitted to each of the millions of cells of living organisms. While DNA sets the stage for human development, it does not write